Hey everyone, welcome back to Biotic World, your one-stop channel for all things biology. Today we are diving deep into one of the most fascinating and ever-evolving fields in science, biotechnology. For revolutionizing healthcare to transforming agriculture, biotechnology is an essential part of our daily lives and the future of science. So, if you have ever wondered how we can use living organisms or their components to solve complex problems, this video is for you. We will be exploring the principles and processes that form the backbone of this exciting field. Let's get started. First things first, what exactly is biotechnology? At its core, biotechnology is the application of biological systems, organisms, and their derivatives to develop or create products for a specific purpose. Think of it as using biology in combination with engineering and technology to make products that improve lives. Biotechnology spans across various industries and it's a driving force in fields such as healthcare, agriculture, food production, and even environmental management. It can be broadly divided into two categories: traditional biotechnology and modern biotechnology. Traditional biotechnology. This includes older techniques like fermentation, selective breeding, and biochemical processes that have been used for centuries to produce beer, bread, cheese, and medicine. Next is the modern biotechnology. This refers to more recent advances like genetic engineering, recombinant DNA technology, gene editing tools like CRISPR-Cas9, and synthetic biology. These innovations enable us to manipulate DNA at a molecular level, creating genetically modified organisms and even design new proteins with specific functions. Now let's break down the key principles that form the foundation of biotechnology. These are the scientific techniques that power everything from creating new medicines to improving crop yields. Genetic engineering is the cornerstone of modern biotechnology. It involves altering the genetic makeup of organisms by manipulating their DNA. This can be done in various ways. Recombinant DNA technology. Here, scientists create and paste pieces of DNA from different organisms to create a new combination of genetic material. This process is fundamental in producing genetically modified organisms that can grow faster, be disease resistant, or contain beneficial traits like improved nutritional content. Next is gene editing. Tools like CRISPR-Cas9 have revolutionized biotechnology by allowing precise changes to the genome of an organism. With CRISPR, scientists can cut specific parts of the DNA and replace them with new genetic material, opening up new possibilities for gene therapies, disease prevention, and even designer crops. Another critical principle is cell culture, which is the process of growing cells in a controlled environment outside of their natural organism. It allows researchers to study cells, tissues, and organs in isolation. This technique is essential in biotechnology for developing vaccines, gene therapies, and pharmaceutical products. One of the most revolutionary uses of cell culture is cloning, especially in the context of somatic cell nuclear transfer. Cloning allows the creation of genetically identical organisms, which is useful for studying diseases and producing genetically engineered animals for research or agriculture. Bioprocessing refers to using living organisms or living components like enzymes or cells to produce goods. Fermentation is one of the oldest forms of bioprocessing and widely used in industries like food and pharmaceuticals. It involves the breakdown of organic materials by microorganisms such as yeast or bacteria to produce products like alcohol, antibiotics, vitamins, and even biofuels. Now that we have covered the key principle, let's dive into some of the critical processes that make biotechnology a game changer. These are the steps researchers follow to manipulate biological systems for specific purposes. First, the gene cloning. Gene cloning is the process of creating copies of specific genes or DNA sequences. First, scientists isolate the gene of interest and then they insert it into a vector like a plasmid or virus to transfer it into a host cell. This allows the host organism to replicate the gene or produce the protein encoded by that gene. This technique is fundamental in producing substances like insulin, human growth hormones, and vaccines. Next is about the PCR. PCR is one of the most powerful tools in biotechnology. It allows scientists to amplify tiny amounts of DNA to create millions of copies. This process is crucial for genetic testing, diagnostics, forensic analysis, and DNA fingerprinting. It allows used extensively in research and disease detection, including testing for the COVID-19 virus. Biotechnology relies on large-scale production techniques, and bioreactors are at the heart of this. In bioreactors, microorganisms are cultured in controlled environments to produce antibiotics, enzymes, biofuels, and other valuable products. The fermentation process involves feeding the microorganisms a nutrient-rich medium that allows them to grow and produce the desired product. Next is about the protein engineering. It involves modifying proteins at the molecular level to improve their properties or give them new functions. This can be done by changing the amino acid sequence of the protein to alter its structure or function. For example, engineered enzymes can be created for industrial applications or designed to treat diseases like cancer. Monoclonal antibodies, which are engineered proteins, are now widely used in cancer treatment. Biotechnology is truly a multidisciplinary field with countless applications. Let's explore how these principles and processes are being applied across industries. First, in medicine, biotechnology has revolutionized drug production, vaccines, and even the possibility of curing genetic disorders. Techniques like gene therapy are enabling the treatment of diseases that were once considered untreatable. For example, gene editing tools like CRISPR are being used to correct defective genes in diseases like sickle cell anemia and hemophilia. Biotechnology in transforming agriculture by creating genetically modified crops that can withstand diseases, pests, and extreme weather conditions. For example, BT cotton and Roundup Ready soybeans are engineered to resist insects and tolerate herbicides, leading to higher crop yield and less resilience on chemical pesticides. Biotechnology also plays a critical role in environmental sustainability. Bioremediation, which uses microorganisms to break down pollutant in the soil or water, is helping clean up oil spills and toxic waste. Additionally, biotechnology is used to produce biofuels, which are renewable and environmentally friendly alternatives to fossil fuels. In industrial biotechnology, microorganisms and enzymes are used to produce bio-based products such as biodegradable plastics, cleaning agents, and food additives. The shift from petroleum-based to bio-based product is an essential step toward a more sustainable and circular economy. As you can see, biotechnology is transforming the world in countless ways. From gene editing to a bio-based product, the applications of biotechnology are limitless, and the principles and processes we have discussed today are the building blocks of this revolutionary field. Whether it's improving healthcare, feeding the world, or protecting the environment, biotechnology is making a profound impact. If you found this video informative, don't forget to like, subscribe.